Okay, so this is a brief overview of the history and the neuroscience of psychedelic therapy. As you heard, Robin wasn't able to join us, so I'll briefly review where we have been and how we've gotten to where James has explained we are. So, first thing to say is that psychedelics have been around before humans, obviously, and they've been part of many human cultures. Perhaps most noticeably, the ancient Greek culture of using ergot and alcohol to fuel insights into the development of phenomena such as democracy. But the real breakthrough in modern times came with LSD, the synthetics analog or derivative of ergot, which was designed to have effects on the vasculature in the brain, but turned out to have powerful effects on serotonin receptors. There's Albert Hofmann, the man who discovered it. And despite using it regularly for 50 odd years, he lived to over 100. And he was so taken with the potential power of LSD as a tool to understand the brain and to elucidate mental phenomena. He persuaded his company, Sandoz, in Basel to make it as a medicine. And there it is, Delicid. It was made available to researchers around the world. In those days, no controls at all, except Sandoz basically said you had to have a level of competence to be able to work with it. And it was used for these purposes to model psychosis, something similar to what Professor Mittal in King's is doing now. That it was used self-experimentation by mental health professionals to give doctors a sense of what it was like to have an altered consciousness. And it was used in two sorts of psychotherapy, the psychedelic psychotherapy, which is a bit like what we're doing today, giving people a high dose, a single trip, produce powerful changes in attitudes and thinking. And psycholytic psychotherapy, a lower dose, given repeatedly to facilitate access to psychological insight. And there was an analysis done in the 1960s following over 130 NIH grants in the USA were spent on LSD research. So, apologies for those of you who couldn't see the screen up till now. This slide shows an analysis of the research that was done into the 1960s. 5,000 subjects in this particular analysis, 25,000 drug sessions, a long-term follow-up of over 20 years of 4,000 patients in the UK. And I think then the key thing is the analysis at the bottom. Treatment with LSD is not without acute adverse reactions, but given adequate psychiatric supervision and proper conditions for its administration, the incidence of such reactions is not great. And it's been argued by one of the pioneers of this research who continued to use psychedelics after they were banned in 1967, Dan Groff. He said psychedelics used responsibly and with proper caution would be for psychiatry what the microscope is for biology and medicine and the telescope is for astronomy. So we had this resurgence, as Jane showed you, massive amount of research in the 50s and 60s, and then LSD got banned. And it got banned largely as a result of the Nixon war on drugs and the fact that the US government believed that it was fueling an anti-war movement, in part championed by Timothy Leary in the middle here. And the drugs got banned. They got banned on the basis of fabrication of lies about their harms. Here are some examples of the lies that were told to justify the banning of LSD. These are clear fabrications in terms of claims that are made, but it was sufficient then, as it is now in many ways, in many countries, for newspaper editors to be concerned about the harms of drugs to enable them to be banned. And what's even more remarkable is that they were banned in the face of opposition from the most powerful man in the world at the time, the man who would have been president if he wasn't assassinated, Bobby Kennedy, who argues with his bureaucrats, why, if these clinical LSD projects were worthwhile 6 months ago, why aren't they worthwhile now? We keep on going around and around. If I could get a flat answer about that, I'd be happy. Is there a misunderstanding about my question? He knew he was being misled by his administrators. And I think, there's an interesting message here because we all know that one of the problems with the drugs being in Schedule 1 is that there is a bureaucratic desire not to change things. And it can be, if bureaucrats can make these drugs illegal, then they can certainly stop them becoming easily available now. And he said, I think perhaps we've lost sight of the fact that LSD can be very, very helpful in our society if used properly. And he was dead right. We wasted 50 years really achieving that. And I would argue that's the worst censorship of research in the history of the world, certainly in relation to anything to do with biomedical science. Well, we've been fighting back now, and there's Robin, a younger Robin, who was not able to speak today, and Amanda Fielding, who, with Robin, helped me set up the Beckley Imperial Research Foundation, which did some of the initial pioneering neuroscience that kind of gave an impetus to resurrecting psychedelic therapy in the UK. And we started off by asking the question, what is this drug, psilocybin, the magic mushrooms here, what does it do? Well, we knew that it actually bound to these receptors in the brain, these serotonin receptors, and the 5-HT2A receptor here is located in the very high-level thinking, emotional areas of the brain, and psilocybin has relatively high affinity at this receptor. So we decided to inject psilocybin into people in a brain scanner, an fMRI scanner, and see what happened. Confidently thinking there would be some activation of the brain as a result of the interesting alterations in perception and hallucinations, etc. they produced. But to our amazement, we just found that there was a switching off of some brain regions. So Leary was wrong. Psychedelics don't turn on the brain, they turn off the brain. But they turn off particularly these control centers, these anterior and posterior control centers of the brain. And by doing that, they allow the brain to work in a more flexible, entropic way, a more open way. And that's illustrated by this beautiful diagram from the maths group at King's, who looked at our data and showed that under psilocybin, there's many, many, many more opportunities for connectivity across the brain. Normally, our brains are very efficient. They do what they've been told to do by decades of education. So they do very much the same thing day in, day out. And this is called the small world brain. But under psychedelics, those pathways that have been laid down forever get broken and new connections can be made. And those new connections can help people, we believe, get new insights into their old problems. And there were two discoveries that we made from this imaging work that pointed towards utility in depression. The first is shown here, and you see in the yellow circle, there's an area here which is another blue, another area of the brain turned down. But this area is a particularly important area in relation to depression because it's called the subgenual cingulate cortex. And many treatments for depression dampen down that area. You see here, all these different treatments dampen down activity in that region. It's a necessary requirement for a treatment to work in depression. And even placebo, if you get better on placebo, you have to dampen down that region. And psilocybin did. So we thought, well, that's very interesting. So maybe it would work in depression. But then the other insight came from the recent discovery of what's called the default mode network, a network that is shown here in the top images in red, the linking here of the frontal and parietal parts of the brain with these lateral parietal regions, so anterior, posterior, cingulate, lateral parietal regions. That network there is called the default mode network, and it's a network which encodes your sense of self. And psilocybin completely disrupts that. You see here, under placebo, it's tight, the frontal and posterior regions are tightly connected, correlated activity, and then under psilocybin, they're disconnected. Well, what's that got to do with depression? It turns out that there's considerable evidence that there's overactivity in the default mode in depression. Here are normal controls, and you see more of the brain is linked in this default mode in depression. And the greater the connectivity, the default mode, the greater the rumination scores. And that makes perfect sense because depression is a disorder in which individuals are thinking excessively about themselves, about mistakes and errors they've made, and feeling guilty, etc. And here you can see that the subgenual cingulate is actually orchestrating this overactivity of the default mode. So if psilocybin disrupts the default mode and the default mode is overactive in depression, that might potentially predict why it would reduce depression. And there was evidence coming from the Hopkins group that in people who weren't depressed, psilocybin could produce enduring improvements in a sense of well-being. And so we argued that perhaps psilocybin would be antidepressant. And we eventually managed to get a grant from the Medical Research Council based on the fact that depression is an enormous problem. The grant was the easiest thing to do. It took 3 iterations to get ethics to approve the use of psilocybin in depressed people. It took 13 months to get the drug supply because of the huge bureaucratic burden of Schedule 1 drugs. And all in all, it cost about £1,500 per dose. And that cost is almost totally driven by the fact that this is a Schedule 1 drug. But we did it as a couple of our doctors in our therapy room. It was an open label feasibility study. It was treatment-resistant depression. And the results were quite remarkable, possibly the most powerful single intervention impact in resistant depression there's ever been. You can see of the 12 subjects, everyone got better. Their depression improved by 1 week. And half of them were completely recovered. They were depression-free at 1 week, and about a third were depression-free at 3 months. So this was an extraordinarily powerful discovery. We found brain changes which correlate with this. And it gave us the impetus to go on and do our own other studies, but also for companies like Compass to set up to try to do this in a more larger, multi-center fashion. And the other thing that became clear from our brain research is that psychedelics work in a very different way to conventional antidepressants. So the SSRIs work in a very focused area of the brain called the limbic system, and it's part of the early emotional brain, and they dampen down feelings. As you heard from, I think, one of the veteran speaking at the beginning, antidepressants dampen down feelings, but they don't resolve the experience. They produce emotional blunting sometimes. Whereas psychedelics work in the cortex, the much higher, more recently evolved part of the brain, and they work on a different receptor, the 2A receptor, and they produce fundamental changes in cognitive processing, cognitive flexibility that allow people to overcome and make sense of their traumas or their depressions and kind of basically escape from them. So these are 2 very different ways now of trying to deal with depression. And if you want to read more about it, there's a little review that me and Robin put together in Cell recently, which goes through the molecular neuroscience of how these drugs work in a bit more detail. So I hope you've made sense to you, and I'm sorry it was a little bit chaotic in terms of starting with the slides. Thank you very much.